Hi, my name is Effie Nikki, and today we're going to talk about a few of the special senses in Chapter 15. Before we start, let's just look at some basic definitions. We should know by now what a receptor is, but a receptor is any structure that's specialized to detect a stimulus. We've seen thermoreceptors before, which detected heat or temperature differences. We've seen nociceptors, which detected pain differences. We're going to see another receptor called a chemoreceptor in just a moment. Again, a sense organ. This is the next definition. If I said to mention your 5 senses or tell me what they are, you could think of those, right? They would be vision, hearing, smell, taste, and touch. We're going to talk about 2 of them today, but it's more than just your nose or your tongue or your ear. It's actual tissue surrounded by nervous tissue, and that tissue is going to enhance the stimulus and relay it to this nervous tissue, and then that's going to travel to your brain so your brain can decide what to do. Remember, your brain's your control center. Okay, so let's now use our fancy anatomy words, gustation and olfaction. So instead of saying taste, we'll say gustation. Instead of smell, we'll say olfaction. I mentioned chemoreceptors. Hopefully we had time to think about it for a second. Chemoreceptors, yes, it's a receptor that detects a chemical stimulus. For taste receptors, they're going to be excited by food chemicals. And for smell receptors, they're going to be excited by airborne chemicals. If you want to think of odor chemicals or odor molecules as well, that would be fine. Here we have the sense of taste slide. We're going to look at some of the taste buds. What are taste buds? They're pretty much receptor organs that are found on the tongue. Let me pull the pen up here. No. There we go. Okay, so we can see the fungiform papillae, and here they're just on the surface of the tongue. The reason they call them fungiform is because they look like mushrooms. We also have folate papillae here on the sides. One that's not mentioned is something called the filiform papillae. Filiform, detects texture differences. So if you have something that's hard and crunchy or something that's soft and smooth in your mouth, you can tell the difference because of these filiform. Let's see. Oh, the major players here. Circumvallate papillae. So in the posterior part of the mouth, kind of looks like an upside down V. These are the circumvallate papillae. This accounts for half of all of your taste buds. We have about 8,000 to 10,000 taste buds, but the majority of the ones are in the posterior portion of your mouth in this circumvallate papillae. So we're going to take a look at these, do an enlarged view. So here we have an enlarged section of a circumvallate papillae. So here's where the half of the circle they've cut. And you can see the individual taste buds and how they all converge onto this nerve. So again, what do? They enlarged another enlargement so that we could actually look at the cells inside a taste bud. So this entire part here is a taste bud. Inside the taste bud, you have gustatory cells. Sometimes they'll be called taste cells, but their technical name is gustatory cells. To me, they kind of look like orange wedges, orange slices. And we know that they're cells because they have a nucleus. What's interesting is that these cells have cilia or hairs, but they're actually microvilli. They're surface extensions. Hopefully you've covered microvilli before now. But what do? They allow you to take in more nutrients and excrete more waste because they've increased the surface area. Okay, so we call them gustatory hairs, and you can see them here, these little extensions. We also have something called a taste pore. This is pretty much an opening where the food molecule is going to have to travel through to get to the gustatory cell. What else? Let's see. Okay, yes, we have the basal cells. What do basal cells do? Basal cells are like stem cells. They make more cells. Gustatory cells regenerate about every 10 days, so you would have to have some sort of stem cell or basal cell to be able to replace those. We've all felt this before. I'm sure we've all burnt our tongue at some time and your taste buds just aren't quite right. You eat something and it doesn't taste like it normally does. Well, that's because you've damaged one of your gustatory or many of your gustatory cells, and they need time to regenerate, and that's what the basal cell is going to do. You can see here too that there are several neurons, and these neurons are actually synapsing with these gustatory cells, and we'll learn more about that in just a moment. But you can see how all of the neurons synapse to the cell and then converge into a cranial nerve. We also have here on the side stratified squamous epithelium. That's all the pink cells. Hopefully we know by now, stratified, many layers, squamous, the shape, epithelium, one of the 4 major types of tissue. Okay, let's move on and actually look at the tongue and look at some taste sensations. There are 5 listed here: sweet, sour, salty, bitter, and umami. Some people will argue that umami is not really a separate taste, that it's a combination of the other tastes, but for this lecture and for your textbook, we're going to list all 5: sweet, sour, salty, bitter, umami. Let's look at their location on the tongue. So here we have sweet, here we have salty and sour, and then in the very back we have bitter. Now, when I write these sensations on the tongue, they're not exclusive to these areas. So it's not that you can only taste sweet on the tip. We have these taste sensations throughout the tongue. This is just where they're highly concentrated. So you have a lot, you're able to taste sweet a lot in the front and bitter in the back. What's interesting about the bitter one, at least to me, they think it's an evolutionary adaptation. Bitter, if you taste something that's really, really bitter, you usually spit it out, right? It causes a gagging or choking or spitting out of the item. A lot of plant toxins are bitter. One of those is arsenic. It's not specifically a plant toxin, it's a mineral, but arsenic is one of those toxins that tastes very bitter, and it's dangerous to us. Arsenic can disrupt cellular respiration at the citric acid cycle. So if you remember anything about cellular respiration, this generates the majority of our ATP. Without ATP, we're not walking, we're not talking, we're not thinking. Our nerves and our muscles have to have ATP to function. So you could see how it would be beneficial that if you put something extremely bitter in your mouth that happened to be toxic, that invoking that gagging response would enable you to spit that item up or not consume more of it and possibly kill you. Okay, let's look at the physiology of taste. So how does a food molecule turn into an action potential? I know that we've covered nerves already, so hopefully I won't have to go through depolarization, threshold potential, action potential. I can just mention the words and you'll know what they are. If not, you can go back and review some of the videos. In order to be tasted, what's going to happen is the food chemical, first of all, it has to be dissolved in saliva. So you're going to have a food chemical or molecule. It has to be dissolved in saliva, and it has to enter the taste pore, and it has to make contact with those gustatory hairs of the gustatory cell. So the food molecule travels through the taste pore, binds to the gustatory cells. The gustatory cells then release a neurotransmitter. That neurotransmitter is going to create what they call a generator potential, and then, or you could say threshold potential. And then if it's significant enough, or you reach over the threshold potential, it'll create an action potential. So again, the food molecule is dissolved in saliva. It comes through the taste pore. It makes contact with the gustatory cells, gustatory hairs. Those that making contact depolarizes the taste cell. So hopefully we remember depolarizing. We're going to have a flood of positively charged ions, sodium, flowing into the cell. That's going to cause a neurotransmitter to be released, and that again is going to create a threshold potential. If the signal or stimulus is strong enough, that will create an action potential. That action potential will go to the brain, and the brain will decide what to do with what the information it was just given or what you've tasted. The next part is showing actually what happens and what parts of the brain does that signal or that cranial nerve travel through, and what does that mean for us and how we taste things. So we can see here we have facial nerve, glossopharyngeal, we have the vagus nerve. These all synapse with the tongue and with these taste buds that we just saw. They travel to the medulla oblongata, then to the pons, then through the thalamus, also through the hypothalamus, and then finally they make their way to the gustatory cortex, which is where your brain, or the seat of taste, or where your brain decides what to do with what you smelled, or excuse me, what you tasted. I'm already on olfaction. Okay, so gustatory cell in the insula lobe. If you remember the insula lobe, you have to take the temporal lobe and the parietal lobe, and you have to retract them to be able to see the insula that's on the inside. Okay, so what's interesting about this? Traveling through the hypothalamus. The hypothalamus can detect and do something about hunger, thirst, hopefully remember some of these. It can increase saliva production. So if you put something in your mouth and it tastes really good and all of a sudden your mouth is just full of saliva, that happened because your hypothalamus made it happen. Also, we travel through an area called the limbic system, if we remember the limbic system. This is your seat of pleasure and aversion. So on the way to the gustatory cortex, that message goes through the limbic system and you decide whether you liked the taste or you didn't like the taste. And then once you get to the gustatory cortex, your brain can say, okay, I'm going to tell the muscles in my mouth to spit that food out because I didn't like it. So I think it's pretty interesting. Hopefully you do too. Okay, let's move on to sense of smell, since I was in such a hurry a moment ago. A sense of smell, what is it, where is it, why do we have to know this? It's really kind of interesting. It's a little bit more difficult. There's a lot more players in olfaction than in gustation, but it's pretty interesting. So let's take a look. We can see here in the diagram, you're actually looking at the nasal cavity. Get the pen working. Here's your nasal cavity. At the roof of your nasal cavity is where you have this olfactory epithelium. We can see the olfactory bulb. The bulb is obviously the part that bulges. The olfactory tract is the part that's going to run to the brain, or the cranial nerve. This is the olfactory cranial nerve 1. We can see a word we should be familiar with from when we did bones, nasal conchae. Remember, we had our inferior nasal conchae, and we had a superior nasal conchae, and we had a medial nasal conchae. The first two were actually part of the ethmoid. So what do you think's back here? Right there. Ethmoid bone. What's interesting is these little projections that hang down, these are neurons. And these are special neurons. They are bipolar neurons that actually have olfactory cilia. This should be very odd to you because we should have never talked about a neuron that has cilia. We've talked about cells that have cilia, but not neurons. So we're going to take a closer look at these in a second, but they're called olfactory receptor cells. Okay, this is a very complicated, detailed, a lot of players going on. I'm going to break it down. We'll look at how all of these work or what they are, and then we'll talk about how they work. But just to put you on the right page, you should be able to see right here the ethmoid. So here's the ethmoid bone, specifically the cribriform plate of the ethmoid bone. So that should kind of give you an idea of where we are. If you want to follow on the next slide, after I go through all the players, if you want to follow the next slide for the actual physiology or how an odor molecule turns into an action potential, you can look at the words on the next page. But first, let's just look through all of these and see if we can make sense of this big mess, right? Down here at the bottom, we have mucus. We're going to have mucus that's secreted by some sort of secretory epithelial cell. Basically, olfactory epithelium. It's going to secrete mucus. We see these supporting cells. These supporting cells have cilia. There's only one row of them. We should pseudostratified columnar or pseudostratified ciliated. We should remember some of those back from our histology chapter. We also have olfactory receptor cell. What is this? This is a neuron. You can see the soma of the neuron. Hopefully we remember these terms. Soma, the body of the neuron. This part right here is the dendrite, and these would be hanging off of the dendrite, and they are cilia. Here we have the axon, and then eventually it's going to synapse with another type of cell. Let's see. Oh, yeah, basal cell. That's what these are, basal cells, the blue cells here. Again, olfactory cells, they're exposed to an external environment, just like your tongue. They're going to get damaged. They're going to have to be regenerated. So you have to have some sort of basal cell or stem cell to create more olfactory receptor cells and more supporting cells here. About every 30 to 50 days, these are going to regenerate. And that's also unusual for a neuron. Neurons don't usually generate that quickly or throughout your life, but these specific olfactory receptor cell neurons actually do. Okay, so let me erase this, and then we can look at how an odor molecule turns into an action potential. So this here is saying the root of inhaled odor molecules. So you're going to have odor molecules here. They have to be dissolved in mucus. They must be. Just like you're in taste, the food molecules had to be dissolved in saliva. Here, these air molecules have to be dissolved in mucus. Now I've just drawn 3 here. When you inhale something or sniff something, a glass of wine, the food in front of you, you're actually breathing in hundreds of millions of odor molecules. So it's not just 1 or 2. This is many, many. So the odor molecule has to be dissolved in the mucus. It has to come in contact with the olfactory cilia. When this occurs, this neuron, this olfactory receptor cell, is going to be depolarized, and that's going to create a threshold potential. If the stimulus is strong enough, that's going to create an action potential, and it's going to synapse here in the olfactory bulb. And then what is it going to synapse with? These mitral cells. Some people say mitral cells, either way. And that is going to converge on the olfactory tract and send a message to your brain saying, I just smelled something, this is what it is. So all of this sensory information from a few odor molecules has to travel through the olfactory receptor cell first, then it goes to the olfactory bulb and it synapses with the mitral or mitral cells. And that's exactly what this slide says. It says dissolved odorants bind to the receptor proteins of the olfactory cilia membrane. What are the receptor proteins? The receptor proteins, you should be thinking, what's that cell? What's the bipolar neuron? So we should be thinking olfactory receptor cells. This creates a second messenger. It can directly polarize this neuron, but sometimes it'll do second messenger. If you don't remember second messenger, you can go back and look at it when we talked about central nervous system and peripheral nervous system. It's just another way to send a signal. You can send the signal directly, depolarize the cell, allowing sodium to enter the cell, or you can have that cascade effect, which happens throughout the cell, and then over here the sodium gate will open. And there was a cyclic GMP. But if you don't remember that, that's okay. Basically, as long as you understand the olfactory receptor cell is the cell that initially triggers the action potential, then it synapses with the mitral cells in the olfactory bulb. And obviously, I have to put a slide in here about the olfactory pathway. This is really pretty interesting. What do these mitral or mitral cells do? They amplify, they refine, and they relay the signal along the olfactory tracts. So where is the olfactory cortex? So remember how we had gustatory cortex, the end spot? Where's the end spot, the olfactory cortex in your brain? It's here in the frontal lobe. I'm just randomly, it may not be exactly there, but it's about there. So this is the first pathway. So these mitral cells will relay the signal to the olfactory cortex and say, deliver the message to the brain. We just smelled fire. So the brain can do what? Oh, well, we need to tell our muscles to coordinate and run. So that's one pathway. The second pathway here, it travels through the hypothalamus, the amygdala, and the limbic system. And hopefully we'll remember some of these. So the hypothalamus here, remember hypothalamus can increase saliva production. So why is it when you smell a steak on the grill, it smells really good and all of a sudden your mouth is watering? That's because this message, the signal from the olfactory tract traveled through the hypothalamus and registered, ooh, that's really great. We need to make more saliva so that we'll encourage ourselves to go get something to eat. Amygdala, this is the seat of memory in the brain, and they show you right here, amygdala. So as this action potential through the olfactory tract, it goes through the amygdala that's being the seat of memory. This is why you can smell a rose and it reminds you of a past boyfriend or girlfriend, or you can smell a perfume, or you can smell chicken noodle soup and it reminds you of your mom or your grandmother. It's because of the pathway that this tract is taking in the brain. And lastly is the limbic system, and we've mentioned this earlier. This is your seat of pleasure and aversion, whether you like it or don't like it. So when you smell something, as that signal or action potential travels through the brain, it goes through the limbic system, and you say, wow, that smells really good. I'm going to go see what that is. Or, ooh, that smells really bad. I'm going to have to stay away from that. The only thing that I failed to mention earlier is that we do have a sense of adaptation with smell. I'm not going to get into the complicated process of how that actually happens, but after a while we become desensitized to smell, and this is actually a good thing. If you smell something too long, your brain is too busy smelling that to be doing other or detecting other stimuli. So we do have that adaptation effect. That's why you can walk into somebody's house and they have 5 or 6 dogs, and to you it smells awful, it smells like dog, but to them it doesn't smell like anything. It's because they have adapted, or their olfactory receptor cells have adapted to that smell. I hope that you've enjoyed this lecture, and I'll see you next lecture. Bye.